Photosynthesis. Let's talk about photosynthesis, the process that plants and bacteria use to convert molecules into carbohydrates that they need to sustain life. We want to concentrate in the photosynthetic process strongly endergonic. That is, it's way uphill thermodynamically. And if you were going to try to use photons to supply the energy for this, well, if you wanted to do it in one photon, that would be a very high energy photon. There aren't a lot of those available from the sun. And somehow we need to use more than one photon for this process. So that's what evolution has done. It's evolved a process of multiple irreversible steps to take photons, convert them into reactive electrons, we'll see, and that will facilitate this reaction up here. Now the next problem is we need a strong absorber of light, but a strong absorber of light is also a strong emitter of light. So how do we efficiently turn photoexcited electrons into redox available electrons before either the electronic excitation simply relaxes by fluorescence or internal conversion, or we lose the electrons altogether? So we know that if we have a strong absorber of light, that once we get up to the excited electronic state, if we have an isolated molecule, that strong absorber will also be a strong fluorescer, and we would lose, through radiative decay, we would lose the energy of the photon. So how can we get around that? Well, first of all, we need a strong absorber. And we know what kinds of molecules absorb well in the visible. Those would be molecules such as keratines and chlorophylls and bilins that have extended conjugated pi-bonded systems. Those are really good for absorbing light. But we got to get around this problem of making sure not only do we have a strong absorber, but we don't want all the light to be fluoresced away. So how do we get rid of that problem? And the answer is that we're going to use a complex system, and we're going to break up the dynamics of this complex system into multiple elementary steps to make photon absorption well separated from photon re-emission. And also we're going to have multiple steps that lead to electron transfer and make it irreversible electron transfer. And the way we're going to do this, of course, is we need to construct some more complex system. Instead of just having isolated molecules, we're going to bind those together somehow to make a light harvesting complex. A chlorosome antenna, it's also called. That's where the light is going to be absorbed. And then we have to figure out a way to turn that excitation into an excited electron that we can deliver to the reaction center. How does that work? Well, this extended complex system is made up of not only the light harvesting complex up here, but that's coupled to this FMO. The FMO is the Fina-Matthews-Olsen protein, illustrated here. It's one of the first proteins to have its crystal structure determined with close to atomic detail. And it's a trimer. Now, of course, we may find out more information on the structure as data always gets better. But at the time of this writing, the structure was known as a trimer. That's what's leading to this highway that's going to somehow transfer our photoexcitation to a reactive electron into the reaction center. Now, each one of these FMO proteins in that trimer there, each one of those has 7 chlorophylls. That's this black molecule here, and they're arranged around that FMO close to each other, about a nanometer apart from each other. So we know the reaction we want to promote, and we're going to do that by absorption of several photons to create these reactive electrons that will help out in this reaction. How are we going to get them there? Well, the first step, of course, is some singlet transition, get us into this excited electronic state. Very quickly, that energy from a localized excitation is going to be redistributed over a number of molecules in that light harvesting complex, and that is an ultrafast process that takes less than 100 femtoseconds. Then there's going to be some way that what we're going to see is we need a number of steps with energy exchange. A little bit of energy is lost in this multiple steps that will take that excitation and move it over to the FMO. This takes much longer, 60 to 70 picoseconds to first delocalize the excitation over several molecules, let it hop till it gets over to the FMO, and then it has to relocalize so that an electron can be exchanged. That's the final process that we have to do, and that takes about 30 to 40 picoseconds once the electron has been transferred from the FMO, finds where the reaction center is. The excitation needs to relocalize, get transferred, and delocalize again in order to become available for reaction. Afterward, of course, an electron has to be returned to the ground state. If it wasn't, then this would essentially stay ionized, this molecule. We don't want that to happen. And it's cytochrome complexes that return the electron, again, in multiple steps, releasing this energy here slowly so that not too much heat is built up in the process. Now, what's going on, right? What we have are a number of these chlorophylls, for instance, here as individual molecules, but they're coupled together, or the molecules in the antenna might absorb a photon anywhere along here, but they need to transfer it to the FMO. Once transferred to the FMO, the excited electron has to find its way to the reaction center where eventually chemistry is initiated. So the problem is a little bit like, you might have seen this game, right? Where the excited electron is going to pop up here at the start and it has to get down there to the finish line, the reaction center. If the excited electron stayed localized, then on its path down here, it could fall back through any one of these holes and be lost radiatively. We don't want that to happen. So what are we going to do? We're going to turn the electron into something that can't easily fall back through one of these holes and relax, and that very efficiently finds the other side. And what we need to do is somehow get the electron not to fit back down the hole and determine what is the best path to the reaction center so that we get a maximum number there. And the way the electron does this is that it's going to use quantum superposition in its excited state. There's going to be a localized excitation that quickly delocalizes. It's going to have to relocalize to find the FMO, and then it's going to go through a similar process to get to the other side, to the reaction center, again localize and be transferred as an excited electron. So the way this looks is a localized excitation, a photon being absorbed by one of the molecules, is rapidly redistributed in the light harvesting complex. That light harvesting complex, right, it's an extended system of coupled molecular species. And so what we like to talk about in that excited state is, it's named an excitonic excited state. It's a collective electronic excitation. That excitation then makes multiple ultrafast inelastic steps, losing a little bit of energy at a time to the vibrations in the LHC. In other words, the electronic excitation is coupled to the phonons, the vibrations of the light harvesting complex, and this is what helps make this process irreversible. You're losing a little bit of energy, so you can't go back to the place where you started and just recombine with the vacancy, the hole that was left behind in the ground state. So that excitation hops along the chlorosome antenna until it finally finds the FMO. You relocalize the excitation into an excited electron. That excited electron is transferred to the FMO, and then a similar process occurs so that the electron is exchanged from the FMO to the reaction center. That's a little bit quicker, 30 to 40 picoseconds. So in sum, Going through this entire process, it takes about 100 picoseconds from the absorption of a photon to the transfer of an electron into the reaction center. And this process, because it's been optimized, all of these coupling events, the formation of the exciton and the coupling to the photons have been optimized such that it's approximately 95% efficient, which is just what a photosynthetic plant or bacteria needs to convert photons efficiently into excitations that lead to photosynthetic chemistry. Now let's look at that excited state in a little more detail. That excited state, this collective excitation, it's composed of a superposition of local states. We know how to form that. A superposition is just a sum over some mixing coefficients times those localized excitations in the molecules. The electronic transition, essentially, right? These are the electronic states of the individual molecules being superimposed on each other to make the collective excitation of the entire LHC, or some small portion of it. Therefore, the transition dipole moment, it's also delocalized over a number of local excitations. Local excitations is an excitation in one molecule. Then the exciton, this excited state, couples to the phonons, the vibrations of the LHC, redistributes the energy, and we get this localized excitation turning into a delocalized excitation. It's able to sample a number of energy transfer pathways, and eventually it gets away from the vacancy in the ground state, so it doesn't recombine with it, right? And will find its way after relocalization into the reaction complex. There's a small loss of energy along the way, but that's a small price to pay because it makes the process irreversible and therefore highly efficient in terms of the number of photons that become excited electrons transferred to the reaction complex.